So hopefully the first two videos have convinced you without a doubt that DNA is in fact the genetic material, better known as the transforming principle. Hopefully I've convinced you of this. So now we are convinced of this and we're going to be like, I wonder what DNA looks like. So here's some clues. So we have nucleic acid chemistry, nucleotides, A, T, C, G, adenine, guanine, cytosine, thymine, uracil. DNA has nucleic acids. They are nucleotides. They are the building blocks of nucleic acids. They consist of a nitrogenous base, two kinds, purines and pyrimidines. We'll talk about those. A pentose sugar and a phosphate. Okay? And it looks like this, right? Purines. So your purines are actually adenine and guanine. So here's your purines, adenine, guanine, right? A purine always binds with a pyrimidine. Okay? See this? These guys always bind with these guys. Yes? Okay. Pyrimidines, these are 6-member single rings. They are your cytosine, thymine, and uracil. Pictured right here. They always bind with one of the purines. Okay, bigger picture. Now, the DNA bases are adenine, cytosine, thymine, guanine. The RNA bases are adenine, cytosine, uracil, guanine. So you're going to replace your T with your U when you go to RNA. RNA contains ribose sugar. What does that even mean? It has a hydroxyl group at carbon 2. Yes, we're going to do organic chemistry. Pause the video, take a deep breath. We're only going to do it for a minute, so settle down. We're going to count these carbons. This is carbon 1, right? Carbon number 1. We're counting carbons. This is carbon number 2. This is carbon number 3, number 4, number 5. We will actually count these carbons a bunch, so just get used to it. 1, 2, 3, 4, 5. Okay? Now, what the difference is here are or RNA has a ribose sugar, which has a hydroxyl group at C2. This is C2, right? DNA contains deoxy without an oxygen. Deoxy, no oxygen. Which one of these contains? Look at this. There's no oxygen there. Look at this carbon 2. There is an oxygen. So this has a hydrogen atom, not a hydroxyl group at H2. Yes? Everyone, hydroxyl group, not a hydroxyl group. RNA, DNA. We good? Everybody good? Deoxyribonucleic acid, DNA, doesn't have an oxygen. Good? Everyone's good? It's organic chemistry. So your eye might start twitching. You might start it's fine, deep breaths. Okay, here it is bigger. Here's some different things about ribonucleosides and deoxyribonucleosides, right? There's a chart. Okay. Nucleosides and nucleotides, they contain they have a nitrogenous base, a pentose sugar, and a phosphate background. Okay, so we have deoxythymine, the TDP, and the adenosine triphosphate. So again, you've got a nitrogenous base and a pentose sugar, nitrogenous base, pentose sugar, and now you have either 2 or 3 phosphates. It's kind of like ADP and ATP. Yes, anyone remember the phosphate groups? Oh yeah. So nucleosides and nucleotides are named for a specific nitrogenous base here. This is the base of which we're going to name our nucleotides. Okay. We also have monophosphates, which is 1 nucleotide, diphosphates, which is has 2 phosphate groups, and triphosphates, which has 3 phosphate groups. So we're just counting phosphate groups. It's not that bad. Everyone settle down. Okay. Triphosphates serve as the precursor molecule during nucleic acid synthesis. That's which one? 3 phosphate groups. ATP and GTP, adenine triphosphate and guanine triphosphate there's a large amount of energy involved in that terminal phosphate group. Right, this is super important in cellular respiration and your ability to breathe. It's all about ATP, and ATP is all about that 3rd phosphate. So just remember, it's all about the 3rd phosphorus. There's a ton of energy here. Okay, phosphodiester bonds. Remember, we're still counting carbons, deep breaths, it's organic chemistry, you're fine. So the linkage between the 2 mononucleotides, 1 mononucleotides. Involve a phosphate group linking two sugars. So it's joining two alcohols by an ester linkage. Let me show you. Calm down. It's not that bad. We're going to count carbons again. Same molecule, right? So we've got carbon 1, carbon 2, carbon 3. This is the 3' end. We will be saying 5' to 3' end like 20 times a day between now and the end, right? This is the 3' end where this little carbon sticks out. Okay, that's the 3' end. So again, let's count them again. 1, 2, 3, 4. Then this guy is 5. This is the 5' carbon attached to this phospho bond. This is the phosphorus, right? This is a phosphodiester bond, which is linked to the 3' carbon from the next one, right? So here's 1, 2, 3, 1, 2, 3, right? And this is how they're linked. This is how they're linked. The 5' is going to attach to the 3'. Yes? Rewind it, take a deep breath. We're just counting carbons. You're fine. And it joins those two alcohols. Do you see how there are two alcohols? See, this is an alcohol, and then this would be, it joins that by an ester linkage. Okay. Phosphodiester bond right here. This is your phosphodiester bond. Two esters, phosphate group, two esters. At the 5' position, and OH group on the 3'. 1, 2, 3, 4, 5. That's where you get that. Oligonucleotides are short chains of about 20 nucleotides. Longer chains are called polynucleotides, and they store vast amounts of genetic information. If you've ever seen all the things that we have sequenced, there's a bunch of DNA there. And this gives rise to extraordinary variation. So again, if you were still not convinced that DNA is a genetic material, it really is. Okay, so back to Chargaff, right? So Chargaff figures out that A goes with T, C goes with G. And he does this by actually looking at the proportion that, hey, if there's a 20% of A's, there's always 20% of T's. And then that gives 80% of C's, right? It's always proportional. So the amount of adenine in any given amount always equals the amount of thymine, and the amount of guanine always equals the amount of cytosine. So he's like, what do you know? A goes with T, C goes with G. So the percentage of C plus G is not always equal to A plus T. You can have 30-30, 40-40, but this always has to be equal. If you have, let's say you have 60% CG, 40% AT, this has got to be 30-30, 20-20. Does that make sense? Okay. Here's this again. Now, let's talk about Rosalind Franklin for 2 minutes. So Rosalind Franklin was she did X-ray chromatography, and she was a woman in a very man field. And she was, the person who ran the lab thought she was hired to be his assistant, but she was hired to have her own lab. He didn't like that. He felt humiliated that a woman was doing this kind of X-ray diffraction. She repeatedly embarrassed Watson and Crick by disproving their theories. So she took this picture. This is called Photo 51. She took this picture using X-ray chromatography to confirm that DNA is a double helix. Without her knowledge, Watson and Crick came in and took this data. And from that, they determined that DNA was a helical structure. The other thing they already knew is Chargaff they already knew about his research that says A goes with T and C goes with G. So that's a lot of evidence pointing to the fact that it's a double helix. So also, Rosalind Franklin, because she had all this X-ray diffraction, got cancer and died at a very young age. So here's what Watson and Crick came up with based on Rosalind Franklin's data, and also from Chargaff's knowing that A goes with T and that it is a double helix and it winds around. Two anti-parallel. A goes with T, C goes with G, right? And you get this? We are going to build some models in class just like this. And here we've got your purines and pyrimidines again. Do you see how this is the molecule that's going in here? We have our phosphodiester bonds. We have paired nitrogen bases. And this is essentially how we get this double helix based on the prior data.